We are very excited to have Dr. Daniel Palacios as our first speaker. Daniel is an associate professor with Oregon State University Marine Mammal Institute. He's based at the Hadfield Marine Science Center in Newport, Oregon, so out on the coast. He's been there since 2013, and since 2020, he's led the Whale Habitat Ecology and Telemetry Lab. Dr. Palacios hails originally from Colombia, and his background is in oceanography. His research addresses questions dealing with large-scale seascapes ecology of top marine predators, including interactions with the environment, with other species, and with human activities. And thank you very much for that bio, Dr. Palacios. And with that, the screen is yours, and thank you.
Excellent. Yeah, good to see you all, virtually, of course, and mostly little squares, but I trust you're all there, and thank you for being here. Thank you, Heather and Victoria, for the invitation. We have been corresponding for probably about the last year or so, and I'm really glad we made it work, and I have the privilege of being the inaugural seminar for this year, 2021. Let's really hope we will be getting together in person soon. So yeah, once again, thank you for the invitation for this seminar series. And yeah, today we're going to be talking about whales. And so you have my background here, and I'm actually wearing my whale socks, which are my good luck socks that I wear every time I give a whale talk anyway. I won't be showing you those, but just trust me, they're pretty cool. They're actually fluoresce in the dark. So really cool. All right, so let me go into presentation mode here. And are you able to see my screen? Excellent. I titled the presentation Here, There, and Everywhere in relation to the movements of humpback whales of the West Coast of the United States. And my reason for doing that is that especially with whale populations recovering around the world, humpback whales especially are turning up everywhere. And you guys, of course, on the East Coast are also having, are having similar situation. And at the same time, encouraging, concerning, yeah. And here out on the West Coast, we have the situation where we have very different populations that come to feed off the West Coast, and that actually poses a challenge for us. So the focus of the presentation really is to give you sort of a broad overview of several research activities that we have been conducting to try and address that multi-population occupation of the West Coast. And I think we have a better idea after this several years of doing this work, but in reality, we're just scratching the surface, and this work really continues. I think what I'll be sharing with you today is the results of sort of a first cut, and this is work that we have been doing funded by the US Navy through the US, the Pacific Fleet Command. And this is not my work alone. I work with a team of great people. There's 4 faculty research assistants in my lab, Barb, Ladd, Thomas, Craig. And I really stand in the shoulder of giants. Bruce Mate was my PhD advisor, and he was my boss until recently when he retired. He's still actively involved, but he's no longer my supervisor. Now Dr. Lisa Balance is our director. But Bruce was instrumental in actually collecting a lot of data that you will see, and of course in developing the whale tags that we have been using. And also we collaborate with the Cetacean Conservation and Genomics Laboratory, Scott Baker and Debbie Steele there. I'll be showing some of the work we're doing with them as well. To start off, I wanted to show a little bit about the background in terms of the humpback whale conservation status at the moment. So in 2016, NMSs revised the Endangered Species Act listing for global humpback whale populations. And rather than managing them as a global endangered population, they separated them into 14 distinct population segments. And that way, the energy and efforts and money can be directed to those population segments that are truly endangered. So there's 4 that are globally listed as endangered and 1 as threatened. And those are in the Atlantic, we have the Cape Verde Island, Northwest Africa. And then in the Pacific, we have the Western North Pacific, the Central America, the Arabian Sea, those are all listed as endangered, and then the Mexico DPS is listed as threatened. Here off the West Coast, we have Mexico, Central America, and we also have a little bit of Hawaii, and we may have every once in a while a straggler from the Western Pacific. So obviously when you encounter a whale off the West Coast, you can't ask it what is your population status. So you have to come up with ways of addressing that. So the second aspect of, let's see what happened. Yeah, there we go. This is a map of those 14 population segments, and they are represented by these circles that are the breeding areas in the low latitudes in the northern and the southern hemisphere. The colors represent the conservation status. So there we have the 4 endangered and the 1 threatened here of Mexico, and the green are the feeding areas. So as I mentioned, we have at least 3 here off the West Coast. Another aspect that's currently ongoing is the designation of critical habitat. Once the species, the populations were listed, the DPSs were listed, then NMS was actually sued to propose critical habitat of the West Coast for these DPSs. So we have also been engaged in that activity in terms of providing NMS with information that is relevant to how they're able to identify habitat that is relevant and that is considered critical for humpback whales. And what you see here is the exclusive economic zone of the West Coast of the United States. And then in Alaska, this is Canada, of course, so we don't have any business there. But in shaded polygons, you see, I believe it's 19 different polygons that are, each of them has a specific consideration in regard to critical habitat for humpback whales, and that's what's being currently proposed. That hasn't completely gone through. I believe just in the last couple of days, they received an extension to when they're supposed to finalize this ruling. One last thing regarding management is that for nymphs on the West Coast, they, within US waters, they manage the stock as a single stock. It's called the California, Oregon, Washington stock. And this map portrays the long-term line transect surveys that have been done since 1991 and the sightings of humpback whales of the West Coast. And as the most recent marine mammal stock assessment, they updated to now recognize that even though it's managed as a single stock, there's actually 2 feeding groups. One that is what's called the California, Oregon feeding group, and the other one, which is the Northern Washington, Southern British Columbia. So we will come back to that because our results point to something that's slightly different from that view, especially for the Oregon whales. So for the rest of the talk, I'll be talking about the work we do with satellite telemetry. And as I mentioned, Bruce Mate was one of the pioneers in developing satellite tracking technology that enables to follow large whales for long distances and long times. So we're talking about complete migration cycles and many months of tracking. And whales, I will say that they are arguably the most difficult animal to track. Most animals that you put a device on, you can handle them or trap them, or you can attach tags to their fur or use collars. Collars are quite common in terrestrial animals. But for any long duration tracking, the only type of device that has worked with any level of success is what we call implant tagging. So what you see, this is a fin whale, and you see just the tip of the end cap, what we call the end cap of the tag. The rest of the tag is embedded in the blubber of the whale, and then we have antenna that transmits the location to the Argos constellation. So this is not GPS, is Argos, which is an entirely different system, and it's based on Doppler shift location. So based on how the frequency that's being transmitted by the tag shifts as the satellite passes overhead. And when you have multiple satellite hits, then you're able to essentially triangulate the location on Earth. And that's what we use. GPS poses, again, whales are just really difficult because they are at the surface for such a short amount of time that you need a two-way communication to get GPS. And although there are some, like FastLog GPS is possible for some of these smaller tags, you just can't use GPS. So we continue to rely on Argos, and I think we will continue to rely on Argos for the foreseeable future. So the tags that we are currently using are tags that Bruce essentially came up with this design in 1997, and we call it the location-only implantable tag. But what's new as of the last few years is that we have been using, incorporating a pressure sensor and a triaxial accelerometer. So we're actually able to collect dive behavior and package it in a way that we can send it via Argos. Yet another challenge is that Argos does not really lend itself to transmission of large amounts of data via satellite. So we have to become very, very ingenious in terms of how we package messages with little pieces of data, which has meant a lot of work abstracting and summarizing dive behavior that we can then download and interpret. So that's all done with software that is able to process. It's called a microprocessor approach. So the software is able to smartly detect the events that we're interested in rather than continuously recording like an archival tag. It's just running a bunch of detection algorithms that pick up on signals that we have pre-specified. And that way we're able to generate summary data for the dives. And right now our three main variables are maximum depth, dive duration, and the number of feeding events. So this is based on very stereotypical lunge feeding behavior that is performed by rorqual whales. If we wanted to use this on a different species that does not lunge feed, we would have to develop a different algorithm to detect feeding events based on the accelerometer. This is sort of a blueprint, a design of the tag. I mentioned the end cap is here, and this is the part that remains external to the whale. And the rest of the components are really inside this integrated or consolidated tag. And this is the anchoring system that allows the tag to stay attached to the whale for several months. And over time, it will start migrating and eventually just falls off the whale. So we do not recover these tags. I'm going to show you a clip of a tagging event in Strait of Juan de Fuca waters in Washington. And this is typical of our operation. So there's 4 people on our RIB, our rigid hull inflatable boat. And so we have a driver, the person on the bow, Sprit here is the tagger. Then the person behind him is me. I'm taking a biopsy sample. And then the 4th person is here, Craig. He operates our cameras. So it requires a lot of coordination and everything happens really quickly. So pay a lot of attention. It has to be a really well-timed, it's a well-oiled machine. It works really well. But we do have earpieces or headsets that we use to communicate, because otherwise the noise of the engine is too loud to be yelling. And essentially the tag is deployed with air pressure, a compressed air applicator. And the tag is what you see up here at the tip, but it's being held by tag holder. So you'll also see the tag holder fly out of the cannon. And for a moment, it stays on the surface. So as soon as we deploy the tag, we stop and pick up that tag holder and pick up also the biopsy dart, which we use to collect a piece of skin and blubber to determine the sex of the animal genetically, as well as nuclear and mitochondrial DNA type of studies. We can also do, we just started working with Seth Newsom on doing stable isotope work. And you can also do hormones with the blubber and lipid contents. So we can actually get a lot out of this one approach that you'll see. So while these two people are super keen on just getting it just right at the right moment, Craig here, he has a handheld camera that he uses to take photos of both the dorsal fin and the flukes of the animal for photo identification. And he's also operating, you see him that he's looking like at a watch. That's actually remote control that he uses to operate 4 cameras. There's one camera right here, another one here. Then this pole here has another camera looking down. And then the camera that is actually collecting this footage is mounted on the shield that is in front of the boat driver. And that's the angle that you'll get to see. So finally, after this long introduction, you're going to be seeing 2 whales, and we're going to be tagging the one that's closer to us. So here we go. And you saw the biopsy arrow sort of fly across to the other side. So yeah, it requires a very close approach, and we take a lot of care in observing the animals for a while from a distance. Make sure that we want to tag nice, big, fat whales. We don't want to tag whales that look skinny or small or anything like that. So we take our time from the distance, just really observing the behavior. If they look like they're calm and they're approachable, then we move in for a tagging approach. Otherwise, we look for other whales. All right. Okay. So stepping back in history a little bit, the tagging technology that Bruce pioneered has been, it had 5 years of sort of development, and he did that work in Hawaii. So what you see here are the tracks of whales that were tagged between 1995 and 2000. That was with ONR funding. And that's essentially some of the very early whale tracking track movements that we have ever seen. And it was fascinating. It was believed that most humpbacks wintering in Hawaii would migrate to Southeast Alaska, but only one of those did, at least completely. Some of the tracks were incomplete. But most importantly, we saw use of the Aleutian Islands, and one whale even went all the way to Kamchatka in Russia. And that was quite new, and it just opened up a lot of questions. Then another study around the same time in, I believe it was 2000. That's to say, yeah, I believe this was in 2000. Some tagging done in the Revillagigedo Islands offshore of Mexico. It's actually quite far offshore of Mexico. And these whales really did not come to the west coast of the US. This whale did come close, but didn't arrive until British Columbia, really. And most of them looked like they were heading toward the Gulf of Alaska. So again, some of the earlier migratory routes for humpback whales in the Pacific Ocean, and also some use of the mainland Mexico breeding area. So that's also some interesting stuff that we're still trying to understand. It's quite complex, this whole situation here. Mainland Mexico, Baja, the islands, and then Central America is very complicated. All right, so I'm going to show you now an update. This is an animation of all our tracking data, and rather than showing it by years, what I'm showing it is by month of the year. So all the tracks are zeroed to start at the first day of the year. So obviously at that time, whatever whales we had tagged were in the breeding areas. And you'll see most of the first tracks here in Hawaii and then Revillagigedo Islands. And then as we go into the summer, you'll see them in the high latitude areas. And then at the end of the feeding season, you'll see the return to the breeding areas. So the colors now distinguish the different areas where we have deployed the tags. So obviously many more tags have been deployed since those early years, especially in the last 4 or 5 years. We have had funding from the Navy and Pacific Life Foundation to do tagging here in Southeast Alaska. You'll see those tags taking off in a moment, as well as off the West Coast in California, especially Oregon and Washington. It's all really exciting stuff. Some tagging done in the Aleutian Islands as well. So now we're into the late feeding season, almost going into the fall. So some southward migration is already going on. So you probably saw one animal went into the Central America. We still have some low numbers, almost not much for Central America. And that's a big question right now because that population is only believed to number about 700 animals and is the one of most concern right now. We also have not yet tagged any animals in the Western Pacific, and that would be very interesting as well. Okay. So now focusing on the West Coast for the remainder of the talk, between 2004 and 2019, we have tracking data for 81 humpbacks, of which 39 are males, 20 are females, and 22 unknown. So the unknowns are because for a variety of reasons, we have not been successful in collecting a biopsy sample at the time of the tagging. And the tags have lasted anywhere from less than a day to 164 days with a mean tracking duration of almost 40 days. By the way, humpbacks, among all the whales we tag of the different species, the tags last the least on humpbacks. And we think that a lot of that has to do with their behavior being so active and acrobatic and generally physical with each other, that the tags just don't last as long. They get bumped out or the electronics suffer. All the breaching, 45 tons of whale landing on a tag, that's gotta be tough. So the maps here show the locations where we have deployed the tags of the West Coast, and we have separated the areas into California, Oregon, Northern California really, Northern California, Oregon, and Washington. Most of the tagging in Washington has been out here in the Strait of Juan de Fuca, at the mouth of the strait. That's a fantastic area for working with humpbacks. And then Oregon offers a different geography, and all of our work has been just day trips wherever we know that there's whale concentration. So it's kind of like up and down the coast, quite the opposite essentially to Washington. And then California, most of the effort has been in Central California, and then just a few tags deployed further south. So then what you see here on the right are the tracks of those animals. And in a way, this map kind of tells a story in that during the feeding season, these animals are generally staying within the areas where they are tagged. Yes, you do see some movement into the adjacent areas, but once it's time to start the breeding migration, they just take off from essentially where they are, and they don't really visit the adjacent areas very much. So the animals that were tagged in Washington waters, they do work up this area of West Vancouver Island and Washington coast, but they don't really come into Oregon very much. And likewise, California animals didn't really come into Oregon very much at all. So that's already given us some idea of some level of distinction or separation of these three areas. There's another way of looking at the same data. Essentially, now I've separated the tracks into those tags that were deployed in Southern and Central California, those that were deployed in Oregon and Northern California, and then those deployed in Washington waters. And the color now represents the time, the month. So you can see that they're spending a lot of time, the whole feeding season, essentially, just kind of like working these areas. And then it was time to go, they just take off. There's a couple animals here in Washington that you see they started on a way to Hawaii before the tag stopped transmitting. And here is an idea of just generally the extent of the movements of these animals during the full tracking period, about 30 degrees. So they all generally span the same area, but once you look at the span in the feeding area alone, with removing the migrating portion of the tracks, we see that animals in Northern Washington are staying fairly restricted, whereas animals in Oregon tend to move a little bit more. So yeah, let's just look at that into a little more detail. So now zooming in a little more, some of these, the polygons that you see are actually Navy areas. So some of this work is, because I mentioned this is being done on behalf of the Navy. So we're interested in seeing how many of the animals that were tagged in California came up to say this is the Northwest Training Range. None of them really, this is actually area W237 of the Northwest Training Range, which is much larger. So just a couple animals really came to the Northwest Training Range, and none of them came into area W237. And then animals tagged in Oregon, Northern California. You see a little bit more of that ranging, of course, they're closer. But yeah, this area is really primarily occupied by animals from Washington. And then likewise to the south, so this is another Navy training range, the SOCAL training range. And yeah, the idea was again to see how much of these areas were being used by humpbacks, and especially in relation to their behavior. So one of the things that you see here, for example, a few animals that were tagged in Oregon, is that once they migrated, they just passed through. So this has implications for management, because animals that are just spending a couple of days transiting through this will be impacted by any human activity or Navy operations. These are areas where the Navy conducts training and testing, will be affected differently if they are just passing through than if they are residing and feeding in these areas. So this is the level of information that we are working up to data to provide in terms of better information that can be used for more targeted management. This map that shows essentially the entire range, the primary purpose is to highlight a little bit of the migration into the breeding areas. So we see that of all the animals that we have tagged, only one has really made it to Central America, which is the population that we think right now is most endangered. We have had several of these animals migrate into Mexican breeding areas, and these are the primary mainland well-known breeding areas. And as I mentioned, a couple of them going to Hawaii up here in Washington. Interesting that some of them follow a migratory route that is pretty far offshore, whereas others stay close to the coast. We tried to look at what's going on there, maybe by sex or by haplotype or something like that. But there's not necessarily a migratory corridor. This is kind of like one of the take-home messages, because some animals, even though once they're feeding or once they're in the breeding area, they're close to the coast. Once they start moving, migrating, they can be far offshore. Yeah, this is a little bit about the timing of the migration, especially for this humpback that we tagged on the return. This was a female that was tagged here in Washington, and then came down fairly coastally, except in the Southern California bight. It just took a wide detour there and then back to the coast. Interestingly, this animal only stayed in the breeding area for 15 days. And then it just returned to the feeding area. So the migration itself lasts about 30 days, more or less, plus or minus. And yeah, these animals, we're seeing the same in Hawaii. They're not really spending a lot of time in the breeding area. So there's sort of a constant parade of animals kind of like coming up and down the coast. It's not like everybody moves together at the same time. Some of them may stay longer. That has to do with age and sex class in terms of how long they stay in the breeding area. Whether an animal has had a calf, then probably a female will stay longer in the breeding area. Yeah, all kinds of interesting questions about how the animals are using these different areas. One other detail is regarding the use of designation that has been in place since 2015. Is not a management designation, but nevertheless, it was promulgated by NMSs. It's called the biologically important areas, and they are 7 of the West Coast for humpback whales. And these were derived from long-term data in terms of sightings and survey data and opportunistic data, not using tagging data. So we had an interest in validating them, essentially using the tagging data that we had, and whether we had any correspondence with what these BIAs are. So yeah, as I mentioned, we have them from south to north. We have Santa Barbara Channel, Morro Bay to Point Sal. The biggest one is this one, the Gulf of the Farallones to Monterey Bay. Fort Bragg to Point Arena. Point St. George, this one is right off at the border between California and Oregon. We only have one in Oregon, Stonewall, Heceta Bank, and then one in Washington, northern Washington. The other thing you see here is the boundaries of the Olympic Coast National Marine Sanctuary. So let's see by tagging region how the whales were occupying these BIAs. So for California animals, the BIAs generally are pretty much spot on, especially this large one, Gulf of the Farallones, Monterey Bay. Yeah, even this one in Morro Bay, it's really great, and Fort Bragg, Point Arena. For Northern California and Oregon, we don't really see much use of these BIAs by these animals. So again, just an idea that these animals seem to be mostly targeting areas that where they generally tend to stay. And if they do wander and move, they don't necessarily end up spending a lot of time in areas that are known or are believed to be important for humpbacks. But then if we move to the animals that we tagged in Oregon and Northern California, then we see, of course, better use. So this is the Point St. George, smaller BIA, and very well used. But we see actually a lot of use outside of these BIAs. So that's one of the conclusions that I'll be coming back later, is that we probably need to be thinking about designation or expansion of some of these BIAs because of what we're seeing with this data. Finally, the Northern Washington BIA. that spot on, looking really good. By the way, this year, these BIAs are due to be revised. They're revised every 5 years. And I believe we're going to be starting working on that. So that's going to be exciting because for the first time, we will be bringing in the tagging data to see how we can better inform BIAs. Yeah, not surprisingly, the larger BIAs have longer residence time. This is in terms of days that animals were inside BIAs. So, for example, you see some very large residence times in the Gulf of the Farallones, Monterey Bay, which is the largest one. But the main take-home message here is that animals that were tagged in Northern Washington had higher occupation of the Northern Washington BIA. The same for animals that were tagged in Oregon. They use this one primarily. And these two, because these two are in Oregon, California. And then obviously the animals that were tagged in California had higher use of the southern BIAs. Not surprisingly, but nevertheless, it's important to get an idea of who is using these BIAs and for how long. So again, this information will be feeding into the new BIA assessment, and it'll be interesting to see where that goes. One other thing we can do with tracking data is to compute home ranges. When you start looking at tracking data for a while, you just kind of like everything becomes like a spaghetti plate. It's just too much, too confusing. So you can derive a home range for each track. And then what we have here is the overlap of home ranges for how many animals home ranges overlap. And the darker the intensity of the color means that more animals are overlapping, and that gives us an idea of the areas of highest use, of highest intensity of use. So not surprisingly, Central California here is very, very important for animals that were tagged in California. And here of Oregon and Northern Washington, I'm sorry, Northern California and Oregon, some areas come up that are interesting. This area here is Stonewall, Heceta Bank, so not surprising. But look at the mouth of the Columbia River starting to light up, as well as Northern California. We do have a BIA here in Point St. George, but this area is much more extensive and it seems to be quite important. And then finally in Northern Washington, generally not any news there, except that the data is essentially illuminating the fact that there is a need for transboundary management, because we are seeing that these animals essentially are using these areas, these waters off Western Vancouver Island quite a bit. Some of them are going inside the inside passage here, the Salish Sea, but by and large, the animals that we tagged, at least, they all made very heavy use of this area. So as it turns out, there is a major shipping lane and also military traffic, vessel traffic going to this area. This hotspot right here is called the Swiftshire Bank. And that's right through the area through which the cruise ships are constantly going back and forth. There's a shipping channel here. And of course, there's Canadian and American shipping traffic going on here. So it's an area that deserves further attention, as I mentioned, transboundary management, because of those issues and the risk of ship strike and noise and all kind of human impacts. I'm going to skip through that. Yeah, this is just one final thing to sort of map, how do the BIAs map onto these areas? Just to give you an idea. And then zooming into the one in Oregon, which of course is my own backyard here. So yeah, I'm based out of, this is Yaquina Bay. I'm based out of the Hatfield Marine Science Center here in Newport. And it's just to see how essentially this BIA probably should be revised, because the use is really a little bit more to the north and maybe even to the south as well. Or animals tagged in Oregon. Interestingly, both animals tagged in California and animals tagged in Washington, they just sort of pass through this area. Additionally, we can do other things. We can do behavioral classification using state-space models, and that model essentially takes the how variable is the heading or the turning angle and the speed to determine whether a whale is in ARS behavior, area restricted search, or essentially patch resident, as opposed to transiting. Those are the two modes that the model is able to separate. And that way give us an idea of the activity the animals are engaged in. So we did that analysis, and based on that analysis, we see that some differences, for example, in Northern Washington, as well as in California, the animals are really spending a lot more time in ARS behavior compared to, say, animals that were tagged in Oregon. Again, the geography of Oregon is different, and the coast and the whole seafloor is very linear. And whereas, say, in California and as well in Washington, you have very convoluted canyon systems, embayments, and that kind of thing. And that leads to essentially probably different prey aggregation types and densities and that kind of stuff, which leads to different foraging strategies. So it's kind of cool that this is coming up in the data. And that also comes up in terms of obviously the speed and distance between each location. They tend to be larger distances between location pairs in in Oregon and Northern California than in Southern Central California or Washington. And likewise, animals move faster. And we looked at things like use by distance to the shelf break, the depth of the seafloor, the slope of the seafloor, distance to shore, aspect of the seafloor. I'm probably not going to go into the details of these things, but if anybody has a question, I'm happy to do that, because we also looked at sea surface temperature and sea surface temperature gradient, chlorophyll for each of these locations where we had tracked these animals. And this is just for the locations where the animals were classified as ARS, or putative feeding behavior. And pretty much all of them turned up significant difference, at least statistically. So how significant or how really different they are, it really deserves a lot more focused work. But nevertheless, it points, again, it's pointing at that there are patterns, that these animals are using the environment in different ways across these areas that up until this point have generally been believed to be part of one single Oregon, California, Washington stock. Moving on to the genetics results. So this is work that is done in Scott Baker's lab. The previous study that did humpback whale genetics was SPLASH. You may have heard of that. It was the sampling, the field work was done in 2004 and 2005. So it's quite old by now. And at the time, there were only 3 tissue samples from Oregon. Now we have 15, still pretty low, but much better than 3. So a couple of things come up because there were only 3 samples back then for the SPLASH project. The Oregon samples were grouped with California samples. And now we have separated them. And what we're seeing is that the samples from Washington and California genetically are very different. Not surprisingly, because most of the animals that use Central California are believed to be part of the Central America DPS, whereas the animals that are using Washington waters are believed to be primarily of the Mexico as well as the Hawaii DPS. So what's happening in Oregon? Oregon is now falling out as significant both from Washington and from California, perhaps not strongly significant, sample size is fairly small. But yeah, I mean, we see a little bit of a mix. So the more blue haplotypes are associated with the Central America DPS, and the more of the maroon and yellow and green colors are more of the Mexico. I think this color here, the yellow one, is actually mostly Hawaii. So yeah, what's happening in Oregon we still don't know, but what we're seeing is a signature that is different. So we just need to do more work out here in Oregon to get more samples. The other thing we can do with the genetics is do a probabilistic assignment to DPS. So for each bar here that you see is one animal, one tissue sample from one animal, and you see at the bottom the number of samples that we have. Actually, we don't, sorry, I had to move my Zoom controls because I couldn't see the bottom. I do not have the numbers here, but just looking at the width of this is all the California animals, these are all the Oregon animals, and this are all the Washington animals. So we have quite a few, like 40 something, 45 or something for Washington, and about 15 for Northern California, Oregon, and then about another 15 or something for Central Southern California. So what the main thing to see here is this, in the probabilistic assignment from 0 to 100 to a DPS, an animal could assign with equal probability to each of these DPSs, or what we see is that most animals really are assigning with highest probability to just one of them. So in California area, the majority are assigning to Central America, which is good, which is what we expect to see, and some to Mexico, which is the yellow, so orange and yellow. In Oregon, we see a little bit of a mix with more animals assigning to Mexico and some animals also assigning even to the Western Pacific. And we're not quite sure what's going on there because, of course, genetics involve inheritance, and it could be, it doesn't necessarily mean that an animal from the Western Pacific was here, especially because these were animals that we tagged and we observed their movements, and we have not seen any migration to the Western Pacific. So it could be more of an ancestral component there. And then finally, once we get into the large Washington sample, we see mostly Mexico, and we're starting to see a lot more Hawaii, especially Hawaii animals. And this is new, we think, since Splash, because the population in Hawaii has been increasing so well, has been growing so well, that they're essentially sort of colonizing adjacent areas. So more of them are coming to Washington waters to feed. And the last thing I have to show you is photo ID work. So when we do our work, we, as you guys saw, we deploy the tag, we collect a piece of skin, and we collect a photograph. So these are photo ID matches using Happy Whale, which is an online resource that does the matching using automated state-of-the-art algorithms. And it's just incredible. You get like 99% accuracy in matching rate, and it does it all for you. So the colors again represent the areas where we tag the animals, and where the arcs and the width of the arcs represents the number of connections, the number of matches to these other areas. So it's really informative to, because we do not, or in other words, say this, we photo ID many more animals than we tag. As I mentioned, as we approach them, we take a close look at how they're doing, and based on a number of variables, we decide whether we want to tag them or not. So in the process, we're collecting a lot of photographs. So our sample size for photo ID is much larger than tagging or biopsy samples. But we see essentially the same patterns, where we're seeing connections to Central America, mostly for animals tagged or photographed in Central California. And then as you start moving further up north into Oregon, then more connections are being made to mainland Mexico and Baja here. And then finally, once you are in the farthest north in Washington, many more connections to Hawaii, some connections to Baja, and some connections to, well, most of the connections really are to Mexico, and then some connections to mainland Mexico. So I hope that I had given you an idea of how really complex the situation is that we're dealing with, and what the new data is helping us sort of better understand. Obviously, there's a lot more work to be done, and a lot more work that we need to do with this kind of data to hopefully put the picture together. So I'm going to stop there, and maybe we have some questions, because I realize we're about 3 minutes away from the hour.
Great, thank you very much. If anybody has a question, please feel free to type it in the chat or to unmute yourself and ask that too, if you'd like.
Thank you for an outstanding presentation. I really appreciate it.
Thanks, Suzanne.
Varied data. I was curious, early on you showed tagged whales heading, one of the whales was heading like a torpedo towards the Aleutians, whereas other whales had more, meandering paths. Can you say anything about that individual?
Yeah, not much really. What we see is a lot of variability in terms of migration speed and even migration route. Whether as the animals get older, they just know their migration much better and they don't necessarily want to be meandering around or anything like that. It's very unclear. And yeah, we just really can't say much about what's happening once you get into the high season. Nobody has really spent much time working in those remote areas. But it could be a number of things. And yeah, I'm afraid that I don't have really good insight into what's going on there, but we do observe that a lot. Some animals just a lot of meandering, and some of them just very, very sure about where they want to go.
It's just always surprising to see a biological process without variability. I mean, it looked like the thing was attached to a glider or something.
Okay. So I think I know what you're talking about. For some of the tracks in that map, because of the infrequency or the irregularity that we get the Argos data, sometimes the tags actually stop transmitting for multiple days. So I believe what you might have seen was a dashed line connecting some very remote locations that may have been apart maybe 30 days or something like that. So that would cause a very, very direct, very straight line, as you're saying. I think that's probably what you saw. Yes, I should probably have pointed that. Thanks for the question, Suzanne.
There's a question from Karen Wishner in the chat, if you can see it.
Yeah, I'm looking at that right now. Thank you, Karen. Do you have birth ID information? Do the offspring of a particular whale go to the same? Yes, so that the genetics are very definitive about that. They have concluded some very strong female philopatry that's driven by the calf where they are born, and then they accompany the mother on the migration to the feeding area. So they actually have both philopatry to the feeding area and to the breeding area, in that obviously when they're born with the mother, that's where they know to be to go to breed. And then they accompany the mother into the feeding area, which means that they will have a lot of fidelity to the feeding area as well. So that is a lot of the driver for humpback whale globally. Humpback whale population structure has to do with that process of strong fidelity both to natal and to feeding areas. That said, we also see variability in that some animals that are, especially the males, some years will go to a different breeding area. We have seen that even with the animals that we have studied, where we have photo ID'd an animal in Hawaii in one season, and then in the following season, we have it has been photographed in Mexico, for example. And likewise for the feeding areas, they will switch feeding areas sometimes, but those are generally very, very few. The bulk of the animals do the same thing, and we actually have tagged animals in subsequent years, and the migration routes are just stunningly similar between years. It's just incredible. Tricia is asking, since your lab has an impressive time series of whale migration patterns, do you have you noticed any changes over time in relation to migration or whale abundance? You have mentioned the Hawaiian population is doing well. Right. It's hard to say, and some of the questions of that have to do with climate change, because one of the first things that people want to know is if migration is being affected by climate change. I don't think I can say that at this point, because what we are trying to tease out at this time is trends. As you well pointed out with this time series, we can start looking at trends that have to do more with climate variability. Out here on the West Coast, we have major events happening with some periodicities like El Nino and La Nina. And then on top of that, now we're having these heat waves. And we are trying to tease out the response to that kind of pattern. And those responses are behaviors, that changes in behavior and movement behavior, types of how much time are they spending moving around versus staying put in one place. That's the information that we are largely focusing right now, because I think we can see a cleaner signal. When you're talking about migration patterns and the long distance migration patterns, that is much harder to get at, much harder, because I believe we need a much larger sample size to really be able to say something about that. So unfortunately, at this point, I don't think we can say anything about changes over time directly with the tracking data, which is the reason why we use multiple lines of evidence to try and put the picture together. I think that photo ID would be, even the photo ID would not be able to show you the actual migration routes that these animals are following, just sort of the two ends, at the two migratory ends. That is a better data source for looking at that question. And even that is somewhat complicated because there's the issue of effort. So if you start looking at, there's photos that go back to the late 70s, but there's very few people who are doing work in certain areas. These days with citizen science especially, there's all kinds of data coming in into resources like Happy Whale. But then how do you tease out the fact that a lot of the lack of data is because there really wasn't a lot of effort back then. So some things you can do, like, maybe restrict the sample to the last 10 years that we believe have been generally well sampled. But then, of course, you lose a little bit of that sort of longer term horizon.
Okay, thank you all very much for coming to Bio Noon. We'll move to the portion with postdocs, a discussion with postdocs and grad students. So anybody stay on the call if you would like to continue the conversation. Thank you all.
Thank you.